All right, so continuing the lecture about Mendel and Mendelian crosses, we're now going to talk a little bit about Punnett squares. So the purpose of a Punnett square is it's an organized way to represent the possible gametes that each parent can make. So you got to remember last chapter when we talked about what a gamete was. A gamete is a sperm or an egg. When an organism goes through meiosis, big T, little t, it's either going to give a big T or it's going to give a little t, and then the other parent is going to give the second copy, right? So what we're representing here, if we take a look, is we're representing that in this cross, if I go back for a second, sorry, we're crossing big T, little t with big T, little t. So each parent was big T, little t, so the first parent could give big T or little t. The second parent could give big T or little t, so this is parent 1, parent 2, and then we fill in the boxes. Big T, big T, big T, little t, big T, little t, little t, little t. We always write the capital letter first. Notice there's two different ways you could get big T, little t. And then our outcome here, the phenotype ratio, in other words, the ratio of what they'll look like, that's the phenotypic ratio, would be 3/4 tall, 1/4 short. Sometimes this is also written as percents, 75% tall, 25% short. And another way this could be written is a 3-to-1 ratio. That means out of 4 total, 3 tall, 1 short. Be really careful because I notice sometimes the student will do this. They'll write 3 to 4, thinking that means the same thing as 3/4. That's not what it means. In genetics, this means out of 7, you expected 3 tall, 4 short, which doesn't make any sense. You would write it like this. So 3-to-1, 3 tall to 1 short. The genotype ratio is the genes. So we would expect 1/4 to be big T, 2/4 or 1/2 big T, little t, 1/4 little t, little t. Again, this can sometimes be written as a 1-to-2-to-1 ratio. Big, 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 little, little, little. Or 25%, 50%, 25%. So percents, it can be fractions, or sometimes you may see it written this way as a ratio. Now, here's the thing. A lot of times freshman year, I notice that we just kind of teach everybody to always make 4 boxes. But remember, the whole point of the Punnett square is just to represent the possibilities of gametes that each parent can make. So if you look at this cross, for example, this parent, if it's big B, big B, they can only make big B. That's it. There's no options. And this parent, big B, little b, can make big B or little b. So you don't need 4 boxes. Will it be wrong if you make 4 boxes? No. But keep in mind, these problems are going to get more complicated, so you're going to save time if you cut down the number of boxes. Here's what it would look like if I make all 4 boxes. Notice, you're going to get the same exact answer. You just end up writing this row a second time. And if you have a fraction, you're going to end up reducing it. So you get the same outcome, 1-to-1 versus 2-to-2, which is the same thing. If you were going to do this cross, and most of you probably would just do it in your head, but if you were going to make a Punnett square, it would actually only need to be a 1-by-1 Punnett square, because each parent only has one option. And therefore, all the offspring would be big B, little b. So you don't need to do this, which I've seen people do, where they do this, and then they fill in all 4 boxes with big B, little b's. Yes, that is 4 out of 4, which is the same thing as 100% or all of them. So just a heads up, you can save time, and you want to do that. Time's not your friend on AP exams, right? You want to work through these problems as quickly as possible. All right, so here are some sample problems. So if tall is dominant to short, what letter should we use? So our rule of thumb was you should probably pick the letter of the dominant trait. Use capital T for tall, use little t for short. I highly recommend you do not use 2 different letters because we're doing 1 gene, height, and then 2 versions of the gene, 2 alleles. The code for a homozygous tall would be big T, big T. The code for a homozygous short would be little t, little t. If we cross a heterozygous with a short, what would be the genotype and phenotype ratios? Okay, so heterozygous, big T, little t. Short, little t, because we know short is recessive. We don't need 4 boxes. You will get the same answer if you use 4 boxes, but you don't need 4. you get this. The genotype ratio would be 1/2 or 50%, big T, little t, 1/2 or 50% little t, or you could also write this as a 1-to-1 ratio of big T, little t to little t, little t. And the phenotype ratio would be that 1/2 would be tall and 1/2 would be short. And again, you could also write it as 1-to-1, or you could write it as 50-50. All right, and then if we cross 2 black rabbits, this is a really good problem to get ready for the test. If you cross 2 black rabbits and the offspring come out 7 black and 2 white, what were the genotypes of the parents? So they want the letters. Well, first of all, if black was recessive, then crossing black should give all black. But since we crossed 2 black and we got some white, we can actually figure out that black is dominant and white must be recessive. Although in class I actually told you to just add that black is dominant just to simplify the problem a little bit. So here's the thing. If we know black is dominant and white is recessive, our 2 black rabbits have to be big B something. Because if they were little b, they'd be white, right? They wouldn't be black. So the fact that our parents are black, we know that they have a big B. The numbers don't really matter here. Don't get tied up in trying to figure out what the 7 and the 2 mean. Here's what's important. They made 2 white rabbits, and if white is recessive, that's little b, little b. Well, the only way these 2 parents can make a white rabbit would be if they each are carrying a copy of little b. So it would have to be big B, little b times big B, little b. That's the only option. It can't be big B times big B, little b, because you can't get little b from that cross, because this parent wouldn't have a little b to give. All right, and then our last problem very quickly, we're crossing a homozygous black and a homozygous white. That's going to be a P generation because they're purebreds. And then cross the F1. That just means cross 2 of the offspring with each other. So I'm going to do it up here. I don't have a lot of space. So the homozygous black would be big B, and the homozygous white would be little b. This is our P generation. You don't really need a Punnett square for this. The F1, they're all going to be big B, little b. That's the F1. When they say to cross the F1, they mean cross these with each other. So big B, little b with big B, little b, we're going to be getting the 4-box Punnett square. It does need to be 4 boxes because each parent can give 2 possibilities. So a 2 by 2 Punnett square. And then they want the genotype and phenotype ratios. So the genotype ratio here would be the 1 to 2 to 1. In other words, 1/4 would be black, 2/4 or 1/2 would be, I'm sorry, that is not a genotype. Big B, little b, and then the last one, the 1/4, little b. That's the genotype ratio, 1 to 2 to 1, or 1/4, 1/2, 1/4, or 25%, 50%, 25%. The phenotype ratio is going to be, this is our F2 in here, just, sorry, I meant to say that. Our phenotype ratio is what they're going to look like. So this is going to be 3/4 black, or 75%, 1/4 white, or 25%, or a 3 to 1 ratio, 3 black to 1 white out of a total of 4 offspring. So that's how you do these problems. All right, test cross. I believe the next lecture actually starts with this, but I'm going to cover it real quick since we have a little time. So the purpose of a test cross, it's sort of a way to work backwards to figure out what the genotype is for an organism that you don't actually know its genes, but you just know what it looks like. So pretend like, we're simplifying here, that cats could only be black or white, and black was dominant. Also pretend like cats have lots of babies, because this kind of a cross really would only work if an organism has lots of offspring. You need a large sample size. So a black cat could actually be big B, or big B, little b. You wouldn't know if since black is dominant, if you didn't have any other information other than seeing that it was black. So what you cross with is a homozygous recessive, a little b, a white cat. In the next chapter, we're going to talk about something called a dihybrid test cross. It's done for a different purpose, but a test cross always crosses with a recessive. You should remember that. So here's what the purpose of the test cross is. If we cross the cat and all the offspring come out black, this would tell us that our original cat must have been homozygous. Again, assume they have a lot of offspring. If they only have 2 offspring, obviously that's not really enough to say that the cat is purebred. And then if the cat was heterozygous, by crossing with a little b, you have the highest possible chance of getting white offspring. It would be a 50-50 chance. And so in this case, if any cats or kittens came out white, you would know the original cat was Big B Little B. So that's called a test cross. It's a way of crossing with a recessive to try to determine if your original organism was homozygous or heterozygous.